Perks of having short height. Okay, so good morning everyone. I am Royani Saha. So my today's talk is sewage sampling at the human-animal interface, how we can integrate participatory surveillance and wastewater monitoring for the One Health early warning detection systems. So this project is part of a global Fluvial project that I worked on, and let's delve into it. So when we think about the surveillance gap, the traditional surveillance systems misses most of their cases, and the human-animal interface is nearly invisible. So because we can see that annually almost 85% of influenza cases never reaches a clinic, and thus we can see a lag of 1 to 2 weeks reporting delay in the traditional systems. And hence, we see another emerging cause is that 75% of these emerging pathogens are zoonotic in origin, which gives us a direction, a route to which we can see where is our next pandemic or epidemic can begin. So what's missing? When we think about that, real-time community signals, reporting, population-level pathogen detection at the zoonotic interface is missing at the current scenario. And One Health surveillance can bring about a collaborative approach where we can see that each system of this whole approach, like human health, animal health, environment, they speak to each other and shared a de-identified data, which can become the earliest indicator of a health threat without you may not require to visit a clinic. So the vision is the participatory surveillance. There was this conference back in Cambodia where they came up with a minimum of 42 parameters, key parameters of human, environmental, and animal health that can help us create a participatory surveillance. Like for human health, it can be a set of ILI symptoms, fever, reporting, just right from the onset. And for animal health, it can be number of species died, unusual behavior, poultry die-offs, where it died, location-based. And for the environment, it can be water contamination that you saw, or wastewater signals, or food safety. And when we talk about signals, we want to make sure that traditional systems like wastewater-based epidemiology and participatory, traditional wastewater-based epidemiology and participatory surveillance, how they complement with each other. So wastewater-based epidemiology is a quantification of the influenza RNA, which is in the sewage solid. And the participatory surveillance is the bidirectional reporting systems where you can receive and transmit your reports. Both of them have their pros and cons. In participatory surveillance, you have the speed. You have the hours from you can report your symptom onset. And the reach, it captures almost 82% of the gray zone. And this gray zone is the time period between your infection and the detection in the population level. And for the wastewater, it's the coverage. It covers all of the demographics, the zip codes in a sewer shed in a wastewater facility can offer. And thus, it gives a confirmatory test for objective viral measurement. And let's take a step back and look into the evidence that we have seen from the study. So the community-reported symptoms consistently preceded the wastewater viral signals, thus validating this dual approach. And if you can look at the first graph, the first image, it tells you the wastewater-based anomaly, which we created. So whenever you have a wastewater signal and it crosses the threshold of 95% CI, it takes as a strike. So think of it as a 2 weeks, and it is detecting the strikes, the number of times it is actually crossing the threshold. And when it reaches the third threshold, that time it marks as an anomaly, just like a baseball strikeout. And for the participatory surveillance, we have taken the 7 weeks median analysis to look at the number of, whenever the cases on a rolling basis, the numbers are increasing, and how they are complementing to each other, both of these signals. And for the findings, as you can see, we have done a correlation analysis, a temporal alignment on how many days it is we can get the early warning detection system, median being 6 to 7 days. So why the community signals lead wastewater? So we have the animal interface in sewage, and we know that sewage or the wastewater, it can pick up any pathogen signals irrespective of its origin, whether it's a bat or if it's an animal or it's human. So, and we know that bat urine can stay in 52 days in the urine. It's detectable in the wastewater, and we have daily cattle. They shed their H5N1 too. So this is from a theory to practice, a very nice framework of the participatory One Health disease detection system, which was based out in Thailand. So there we can see that the community workers, they were actually able to send reports to these systems, and they can actually report the animal die-off within the hour and what caused that. And this system now spans across multiple countries like Thailand, Tanzania, and this digital app collects human, animal, environmental, all reports in one place. So again, H5N1, another wastewater as a One Health sentinel, another example. In wild birds, the first H5N1 clade was detected. Then we can see that in the dairy cattle, first in March 2024, it was first detected. Then for human cases, it started to rise for 66 plus cases. And thus in the wastewater, we can identify in almost 24 states that H5 subtype was detected. So which brings us together to this One Health integrated surveillance, connecting the pod model and the wastewater evidence, and we can create a One Health integrated system where we can fuse the community signals, we can expand it to animal wastewater streams, we can expand the breadth to different pathogens, and thus we can identify all those zoonotic hotspots and deploy. This will be it. Thank you.